السلام عليكم ورحمة الله تعالى وبركاته. Let me start with introduce myself. My name is محمد. I am lecturer at University of الانبار College of Dentistry. I have got my master and PhD degree in molecular biology. Already, this is the second lecture actually. The first lecture was just introduction of the biology, was the biology, and the branch of the biology, how many, we have a lot. Already you have taken that in the first lecture. If you don't have any idea about that, you can go back to the first lecture, because this is the second lecture. Actually, this lecture focused on the structure and function of the micromolecules. In the first lecture, we have taken the level of the organization, and we have mentioned that the microorganism consists of the atoms, and these atoms will connect to each other to form the molecules, and then these molecules will connect to each other to form the macromolecules. Now we have to after this is to have to answer this question after finish this lecture. What are the macromolecules? Actually, we have 4 types of the macromolecules, and the macromolecules are the polymers of the monomer. Monomer, that means single, poly, that means more than one, di or poly. Monomer subunit linked together by covalent bond via the dehydration reaction. That means after every single reaction, you have to take off the one molecule of water, H2O. Some of references actually talking about, we have just 3 major types of the macromolecules, and some of them talking about 4. Now we are going to mix between them. I am going to talk about the 3, and the other one will be the last one. The first one is the proteins. Proteins composed of amino acid linked together by peptide bonds. And the second one is nucleic acid. The nucleic acid composed of nucleotide, RNA or DNA, linked together by phosphodiester bond. All these bond is the covalent bond. The third is polysaccharide. Polysaccharide, that means composed of monosaccharides, sugar, linked by the glycoside bond. This is some of reference talking about just the 3, and the others talking about the phospholipid or fatty acid or lipid. Phospholipid, the 4th major chemical building block, assumable no equivalent into the biomembranes. This is the difference between the 3 links. All the protein, nucleic acid, and polysaccharide linked together by covalent bonds, and the phospholipid or the fatty acid linked between them by the non-equivalently linked. Now let me start. How can I link monomer to each other? For example, here we have monomer in amino acid. The smallest unit in the protein is called amino acid. When linked to each other, actually the structure of the amino acid we have C here. We have the carbon number 1 here, and we have amino group H2, and we have H group, and carboxy group as well, COH. Here it's called R chain. This will be changeable. That means, for example, here we have 20 types of the amino acid. That means every single amino acid has the different group here to form some amino acid, from 20 amino acid. We can see here in general, for example, here when interact to each other, we can see here red square, here OH, hydroxyl group, and hydrogen group. When interact to each other, the water will take off from this interaction. This is called dehydration reaction. And this after interaction will call polymers. And polymers will give us the proteins. The same things with the nucleotide. Nucleotide, this is called a peptide bond. The interaction between an amino acid or protein called a peptide bond. In a nucleic acid, actually, here we have hydroxy group here and hydrogen group here. This is in 5' and this is 3'. When interact to each other using the phosphodiester bond here, phosphodiester bond will give us the nucleic acid. Will give us nucleic acid, as well as this is dehydration reaction, because the hydrogen molecules will take off from this reaction. The same situation as well with the monosaccharide or the sugar. We can see here this is a glucose. When interact with the glucose using the glycoside bond, this is polysaccharide. Dehydration reaction, this is will give us chain of the glucose. The fourth one will be here. All these reactions, as I mentioned before, all these reactions will be dehydration and as well as covalent bonds. That means the peptide bonds and phosphodiester bond and the glycoside bond is the covalent bond. But here the interaction between the polar or hydrophilic, headed group, and the hydrophobic. The hydrophobic, this is will like the water, while the hydrophilic, this is like the water. We can see here in this head we have two types of the group. The first group is the phosphate and the second group is the glycerol, and the tail consists of two types of the fatty acid. One of them non-saturated and other one is saturated. These just example for the lipid. Now, why the polymers is quite important? Why the, for example, protein is quite important? Why the sugar? Why the lipid, protein, nucleic acid is quite important for the organisms, either human, animal, plants, microorganisms? Was quite important because there is important job to do by these molecules. For example, storing information. We can use, for example, DNA to store for information, as well as catalyze chemical reaction. As I mentioned in lecture 1, we have catabolism and anabolism and metabolism. All these reactions can do it by the proteins or lipid or carbohydrates or others. As well as maybe I gave structural elements that define cell shape and movements and many others. Metabolism, I mentioned metabolism as well. Now, let me start with the carbohydrate. What's the meaning carbohydrate? The chemical structure of carbohydrate. For example, the carbohydrate is the biomolecules, consists of the first one carbon, the second one hydrogen, the third one is oxygen atoms. In general, actually, there is some of the expression used usually with the hydrogen-oxygen atom ratio of 2 to 1, like water exactly. And this with the empirical formula. This is formula of the carbohydrates. We can see here carbon with the M, H2O by N. We can see here where M may be different from N. It's different, M and different. But in general, we can see here the formula holds true for the monosaccharides. Some exception exists. For example, I mean this is not formula for the whole monosaccharide or for the whole polysaccharide, but there's some of the exception. For example, here disaccharides. Already we have, it's like, for example, has to be here H2O5 according to the formula of the water. But here we have some of the different in structure for the deoxyribose. Deoxyribose is quite important for the DNA. Now, the monosaccharide actually, what's the meaning of the monosaccharide? The monosaccharide is the smallest units to form the sugar or carbohydrate. For example, glucose, ribose, deoxyribose, all these are monomers. Monosaccharides are the simplest carbohydrates, and that they cannot be hydrolyzed to the smaller carbohydrates. Cannot. As well as monosaccharides are important fuel molecules as well as building blocks for the nucleic acid, because we have deoxyribose and as well for DNA and ribose in RNA. The smallest monosaccharide, for which N3, for example, dihydroxyacetone and D and L glyceraldehyde. The structure of glucose is quite important. Here we can see here we have 6 atoms of the carbon, and we have oxygen group and hydrogen group and hydroxyl group here and H group here, and as well as we can see this is the structure of the glucose. Glucose so important for our body because they give us the energy for ATP during the cell respiration in nearly all type of the organisms. The glucose is transported to the cell by the insulin by bloodstream in our body. But in plants and bacteria may be different. Not maybe different, definitely will be different. Ribose and deoxyribose are significant. Why? Because they are found respectively in the nucleic acid, RNA, and the DNA. As we know, the DNA has the genetic code for metabolism for the how can I build protein, how can I build carbohydrate, how can I, all this information has to take from the DNA before go to the cytoplasm to translate it to the protein, carbohydrate, or lipid, or etc. We can see here ribose, 5 atoms of the carbon, we can ribose, and here we can see deoxyribose because we lack of the oxygen atom here in group number 2 or atom carbon number 2. Now, what's the meaning of the disaccharide? The disaccharide will join 2 monosaccharides to each other, like sucrose, lactose. Lactose found in milk. When you, for example, when you drink the milk, we have lactose, a lot of lactose in milk, and milk consists of the 2 joint monosaccharide called disaccharide, and these are the smallest polysaccharide. And we can see here the galactose and the glucose will compose the lactose, while the sucrose, for example, this is sugar when you eat it, when you put it in the tea and dissolve, this is composed of glucose and fructose, and when integrated, this is will give you the glucose and the fructose, and the glucose. Actually, all these monosaccharides or disaccharides, all these things will take it from the plants, because we cannot produce or we cannot synthesize the sugar in our body. All these will take it from other source. By the, for example, in plant photosynthesis, and bacteria maybe can use plastida to build the glucose or fructose or other sugars. In general, the formula of disaccharide is, we can see here, C12H22O11. Okay, that means this is the formula of the disaccharide. Now we can see here how can galactose and the glucose interact to each other to give us a lactose, as we know. This link or this bond called galactoside used to link to each other. We can see here we can differentiation between them, and this is beta and this is alpha. Both of them, if you can see to the structure of the glucose and the galactose, the same, but the difference between the just direction of the hydroxy group, OH group. This is, we can see alpha and this is beta. When interact with each other, the dehydration reaction will give us the sucrose here and lactose here. The polysaccharide will be different because we'll join the most of the monomolecules to each other using the more than 2 molecules. For example, glycogen is a glycogen found in the animal and in the human as well. Cellulose are the polymer. All of glycogen or cellulose in the plant and glycogen in human consists of just glucose linked to each other. Glucose branch molecules, or very long molecules. The different type of the chemical bond. Starch as well we have in the plant, and glycogen in animal are used to store energy. That's used to store the energy, as I mentioned before. The glucose is quite important for our body to use it as the energy. In insect, actually, we have chitin. This is used for to build the shield and to build the skeleton for the insect, and in fungi as well. We have taken the first one, carbohydrates. The second one is the proteins. The proteins is quite important in our body. Why? Because the protein, organic molecules, using for example, to build hormones, enzyme, and others as well. For example, how can I use the protein in the cell membrane? How can we transport some of the elements and molecules through the cell membrane? The proteins may be structural, or maybe regulatory, or maybe as well as a protective, to protect the animal, or protect the human beings. They may serve in the transport. We can use it in the transport, in storage, some of the energy, or in cell membrane. Or maybe some of the protein will be toxic, very toxic, like the protein that's secreted by snakes. When you bite from the snake, this will be very, very, very harmful and very dangerous when you go to the bloodstream, and this will cause some of the, maybe lead to the die. In general, we have 20 different chemically distinct amino acids to consist of the polypeptide or proteins. And the amino acid can be in any order, to give you a lot of the type of the proteins. For example, proteins can function and enzyme or hormone. All these will be joined to each other to give you either, for example, either support protein or functional protein or enzyme or hormone and others. Now, in general, the amino acids, as I mentioned before, consist of the hydrogen group, carboxyl group, and H and R group as well. You can see here, this is a carbon group, and this is amino group, R chain, hydroxyl group, and hydrogen group. When interact to each other, dehydration will give you the poly chain. For example, the hormone are chemical signal molecules, usually protein or steroids maybe, secreted by the endocrine gland or group of the endocrine cell that act to control or regulatory specific physiology process. That's mean why the protein is quite important or so important, including growth, development, metabolism, and the protection. For example, insulin is the protein hormone that maintains blood glucose level. If you lose the insulin either by diabetic number 1 or diabetic number 2, this will lead to the serious disease. This is called diabetic mellitus. If you lose the, for example, hormone, insulin hormone, this will lead to the diabetic mellitus. In general, we have function of a protein in human are, for example, metabolism, mentioned that before, support, like nails, like hair, like skin, for example, as well, keratin, make up hair and the nails, and as well as the transport. Some of the protein using as the car when to transport the protein, or for example ions from outside the cell to inside the cell, the supporters in the plasma membrane, as well as other protein transport molecules in the blood, hemoglobin, carry the oxygen to the tissue and to the cells as well. The veins. In our body we have a lot of protein act as the defense. For example, antibiotic. Antibiotic is the proteins act to destroy it, or to fight, or to attack the virus, to the bacteria, or to any foreign body inside our body. So they act as the defense as well. Nucleic acid. The nucleic acid actually is quite important because all information we need to for example, to lipid, to the protein, to the nucleic acid itself, as well found in the DNA, found in the nucleic acid, as well as without RNA I cannot translate the information in nucleic acid to the lipid, to the protein, to the carbohydrate, to the other micromolecules. That means the nucleic acid store all information we need for the catabolism, anabolism, and metabolism in our body. DNA is deoxyribonucleic acid, is one of the type of the nucleic acid that not only store information but how to copy as well, how to copy or replicate itself, but also specific to the order in the which amino acids are to be joined to make the protein. The job for this to the RNA. RNA, ribonucleic acid, is another diverse type of the nucleic acid that has multiple uses. Actually, we have a lot of the types of the messenger RNA. For example, the first one, this is the very copy of the gene in the DNA that is specific what the amino acid sequencing will be during the process of a protein synthesis. Transferred RNA is also necessary in synthesizing the protein and help to translate the sequencing of the nucleic acid in the gene into the correct sequencing of amino acid and polypeptide during the protein synthesis. The ribosomal RNA, it works as the enzyme to form the peptide bond between the amino acid and the polypeptide. Can you see? The RNA is quite important. The ribosome will have joined the amino acid to each other to build the polypeptide and then amino acid. As we know, when after the join to each other, water molecules will leave this reaction, and this reaction called dehydration reaction. The chemical structure of the DNA RNA is you can see here phosphate group, and see here deoxyribose sugar and nitrogen base. This is the structure of the nucleic acid. Here, as we know, we have two types of the bond. We have chemical bond and the physical bond. Chemical bond like covalent bond and the ionic bond. But here, how can the two strands of the DNA interact to each other? Actually using the hydrogen bond. The hydrogen bond, as we know, between the hydrogen and other atoms. So the links between them is chemical, is not is physical, is not chemical. Is physical. Hydrogen bond is physical interaction, not chemical interaction. We can see how adenine here and thymine interact to each other using two hydrogen bonds, and we can see here between the guanine and cytosine using the three type of the hydrogen bond to interact to each other. how can join to each other using the guanine acetylcysteine. The fourth one is the lipids. The lipids is too important actually. Without lipids we can't live. Fat and oil is used by the animal, human as well, for the both insulation and long-term energy store. This energy store for the lipid. It contains two types of subunit molecules, fatty acid and glycerol. Fatty acid and glycerol. In fatty acid we have two types of saturated and unsaturated molecules. For example, saturated fats and unsaturated oil like olives. And phospholipid as we have phospholipid as well. For example, this is an example of the fatty the lipid, phospholipid found in the cell membrane that surround the cell, consists of the phospholipid bilayer, not just one, bilayer. Next lecture will take it how can chemical structure and structure of the cells, cell membrane. Because the head of each molecule is hydrophilic, or you have taken this information, we have two types. We have hydrophilic and hydrophobic region. The phospholipid bilayer is key compound used to keep cell and the biological compartments within cells separated. Okay. The plasma membrane is absolutely essential to the structure and function of the cell, and this synthesis and important to the phospholipid to living things. Actually, the phospholipid is not just to structure and function of microorganism for the cell, but as well as to transport and to support and to as well as like fingerprint. For example, we have markers on the cell membrane help the other cell to identify this cell is belong to our body or maybe no. Maybe all this information will take it in next lecture. Yeah, actually you can see here, this is already I have, you are familiar with this image because already you have shown this image. We have polar group, phosphate group. This is polar group and phosphate group we have, and glycerol group interact with the unsaturated and saturated fatty acid. This is a piece of the cell membrane. A piece of cell membrane. We can see phospholipid bilayer. We can see as well steroid. For example, cholesterol is essential component of the animal cell plasma membrane. We can see the steroid or cholesterol. More specific, cholesterol, we can see cholesterol inside the cell membrane. Actually, this will provide rigidity and as well as physical stability for the cell membrane. And as well as cholesterol will give us several jobs, like we can find cholesterol in sex hormone, either in testosterone in testis, in male, and in female sex hormone, estrogen in ovaries. The last thing in the lipids, we can see boxes. This is we can see in our ear when you after have a shower and you need to clean your ears. After that, we can see this is yellowish material. After this yellowish material consists of cholesterol and consists of the fatty acid and others. Actually, this is quite important because we can stop some of insects to enter our body and sometimes maybe kill it, like ants. Sometimes we can, for example, we can see some ants in the first, especially in the child, we can see killed because this is very toxic and act as the traps for the dust and the insect at the same time. This material as well as to prevent any contamination with the for example, some of the bacteria or some of fungi, is always stop them because it's very important to prevent any infection or inflammation in our inside our ear. Thank you for your listening. والسلام عليكم ورحمة الله تعالى وبركاته.